Cancer is one of those topics that tends to produce two very different reactions when people talk about prevention through diet. Some dismiss the whole idea as wishful thinking, assuming cancer risk comes down almost entirely to genetics and luck. Others swing the opposite direction, treating certain foods as if they were magic bullets, capable of guaranteeing protection on their own. The actual science sits in a much more interesting and, frankly, more useful place between those two extremes. Large-scale population studies, laboratory research on cellular mechanisms, and increasingly well-designed clinical trials have identified specific foods and dietary patterns that are consistently associated with meaningfully lower rates of certain cancers. None of them work as a guarantee, and none of them replace proven medical screening or treatment. But the evidence behind several specific foods has become substantial enough that major cancer research organizations now include them in their formal dietary guidance. Today we're walking through 5 of the most well-supported of these foods, what the research actually shows, and importantly, why they seem to work the way they do at a biological level. Before diving into specific foods, it's worth understanding briefly why diet has any relationship to cancer risk at all, because this context makes the rest of the discussion far more meaningful than simply memorizing a list. Cancer develops through a complex, multi-step process involving damage to the DNA within cells, damage that can come from many sources including environmental exposures, certain infections, and normal cellular processes that occasionally go wrong, even without any external cause. The body has extensive natural defense systems against this process, including antioxidant systems that neutralize damaging molecules called free radicals before they can harm DNA, immune surveillance that identifies and destroys abnormal cells before they can develop into tumors, and various repair mechanisms that fix DNA damage when it does occur. Chronic inflammation, a persistent, low-grade immune activation that doesn't resolve the way inflammation normally should, has emerged in research over the past 2 decades as another significant contributor to cancer development, since the same inflammatory processes involved in fighting infection can, when chronically active, actually promote the kind of cellular changes that lead toward cancer over time. What researchers have found is that specific compounds in certain foods appear to support these natural defense systems, whether by providing antioxidant protection, reducing chronic inflammation, supporting healthy hormone metabolism, or in some cases directly influencing the behavior of cells in ways laboratory research has found relevant to cancer prevention. This is the general framework through which each of the 5 foods we're about to discuss appears to exert its protective association. The first food, and one of the most extensively studied in this entire field of research, is cruciferous vegetables, a category that includes broccoli, cauliflower, cabbage, Brussels sprouts, kale, and bok choy, among others. What makes this category particularly interesting from a scientific standpoint is that researchers have identified specific compounds unique to these vegetables that appear to explain much of their protective association, rather than the benefit simply being a generic effect of eating more vegetables. Cruciferous vegetables contain compounds called glucosinolates, which when the vegetable is chopped, chewed, or digested, are converted by an enzyme into biologically active compounds, including sulforaphane and indole 3-carbinol. Laboratory research on these specific compounds has found that they appear to influence several processes relevant to cancer development, including supporting the liver's ability to metabolize and clear potentially harmful compounds from the body, reducing inflammation, and in cell culture studies, even influencing the growth and survival of abnormal cells in ways that suggest a genuinely protective mechanism rather than a coincidental association. Population studies examining cruciferous vegetable intake have found associations with reduced risk of several cancer types, with some of the more consistent findings involving colorectal cancer and lung cancer, where multiple large studies across different populations have found that higher cruciferous vegetable consumption correlates with meaningfully lower cancer incidence, even after researchers statistically accounted for other lifestyle factors that might otherwise explain the association. Research on sulforaphane specifically has expanded considerably in recent years, including some early-stage clinical trials examining its effects directly. And while this research is still evolving, The consistency of findings across both mechanistic laboratory studies and large population studies has made cruciferous vegetables one of the more confidently recommended food categories in formal cancer prevention dietary guidelines issued by major research and public health organizations. The second food is tomatoes, and specifically the compound they're most known for in this area of research, lycopene, a pigment responsible for tomatoes' red color, and a member of a broader family of protective plant compounds called carotenoids. What makes tomatoes a particularly interesting case study in nutritional science is the well-documented finding that cooking, which normally reduces the nutrient content of vegetables, actually increases the bioavailability of lycopene, meaning the body can absorb and use more of it from cooked or processed tomato products, like tomato sauce or paste, than from raw tomatoes, a finding that has been consistently replicated in research measuring lycopene levels in the bloodstream following different forms of tomato consumption. The research on lycopene and cancer risk has focused most extensively on prostate cancer, where multiple studies, including some larger and longer-running ones, have found associations between higher lycopene intake or higher measured blood lycopene levels and reduced prostate cancer risk. Laboratory research examining lycopene's mechanisms has found it to be a particularly potent antioxidant, capable of neutralizing certain reactive molecules more effectively than some other carotenoids studied, and some research has also found that lycopene appears to influence hormone-related pathways relevant to prostate cell growth, though the exact mechanisms remain an area of active scientific investigation. It's worth noting that while the prostate cancer research is the most extensive, some studies have also examined associations between tomato and lycopene intake and other cancer types, including certain digestive tract cancers, with generally though not universally supportive findings, reflecting the reality that nutritional research often shows stronger and more consistent associations for some cancer types than others, even for the same food. The third food is fatty fish, particularly varieties rich in omega-3 fatty acids like salmon, mackerel, sardines, and trout, and the research here connects to a somewhat different mechanism than the antioxidant-focused compounds we've discussed so far, centering instead on these fatty acids' well-documented anti-inflammatory properties. Given what we discussed earlier about chronic inflammation's role in cancer development, foods that measurably reduce this inflammatory burden throughout the body represent a genuinely distinct and complementary protective pathway compared to foods working primarily through antioxidant mechanisms. Multiple studies have examined the relationship between omega-3 fatty acid intake, whether from fish consumption or in some research from supplementation, and cancer risk, with the most consistent findings emerging in relation to colorectal cancer, where several large population studies have found that higher fish consumption, particularly of fatty fish specifically rather than fish in general, is associated with meaningfully lower risk. Some research has also examined breast cancer risk in relation to omega-3 intake, with findings generally, though not entirely uniformly, supportive of a protective association, and researchers studying this connection point to omega-3 fatty acids' documented ability to reduce production of certain inflammatory signaling molecules, alongside some laboratory research suggesting these fatty acids may directly influence the growth signaling pathways involved in some cancer cell types. It's worth noting a nuance the research has identified here. The association appears considerably stronger for fish consumed as whole food rather than for isolated omega-3 supplements, with several major clinical trials testing fish oil supplements, specifically for cancer prevention, producing more modest or mixed results compared to the observational research on actual fish consumption, a pattern that has led many researchers to suspect that other beneficial compounds present in whole fish, or possibly the way fish consumption often correlates with an overall healthier dietary pattern, may be contributing to the stronger associations found for the whole food compared to the isolated supplement. The fourth food is berries, a category that includes blueberries, strawberries, raspberries, and blackberries, among others, all of which are particularly rich in a class of compounds called polyphenols, and specifically a subgroup called anthocyanins, the pigments responsible for their deep red, blue, and purple colors. Berries have attracted substantial research interest, partly because they contain some of the highest antioxidant concentrations, as measured by standard laboratory assays, of any commonly consumed food category, and researchers have spent considerable effort trying to determine whether this laboratory-measured antioxidant capacity translates into meaningful biological protection when berries are actually consumed as part of a real diet. Research examining berry consumption in relation to cancer has found some genuinely interesting and specific results. Laboratory and some early-stage clinical research has examined black raspberries specifically in relation to esophageal and colorectal tissue, finding that compounds within these berries appear to influence markers of DNA damage and abnormal cell growth in ways that suggest genuine protective potential, though this remains an area where more large-scale human research is still needed to fully confirm the degree of protection in real-world outcomes, rather than laboratory or early-stage measures alone. Broader population studies examining overall berry and polyphenol-rich fruit consumption have found associations with reduced markers of oxidative stress and inflammation throughout the body, and some studies have found associations between higher flavonoid intake broadly, a category that includes the anthocyanins in berries, alongside related compounds found in other plant foods, and reduced risk of certain cancers, though researchers are careful to note that isolating the specific contribution of berries alone, separate from an overall diet that tends to be higher in various protective plant compounds, remains scientifically challenging, given how people's real-world diets naturally cluster different healthy food choices together. The fifth food is green tea, which stands out somewhat from the other items on this list, because unlike whole foods like vegetables, fish, or fruit, it represents a beverage whose primary studied compounds, a group of polyphenols called catechins, with one called EGCG receiving particular research attention, are consumed in a fairly concentrated and consistent form each time it's prepared, making it in some ways easier to study with precision compared to foods with more variable compound content. Research on green tea and cancer risk has been extensive, particularly in populations primarily in parts of Asia, where green tea consumption has historically been high enough and consistent enough to allow researchers to study its relationship with cancer rates across large populations over long periods. Multiple studies have found associations between regular green tea consumption and reduced risk of several cancer types, with some of the more researched associations involving certain digestive tract cancers. Laboratory research on EGCG and related catechins has identified several plausible mechanisms for this association, including antioxidant activity, effects on cell signaling pathways involved in abnormal cell growth, and some research suggesting these compounds may influence the activity of enzymes involved in processing potentially harmful substances within the body. It's worth being appropriately measured about this research since the strength of association varies considerably across different cancer types and different populations studied, and some of the research showing the strongest associations comes from populations with lifelong, high-volume green tea consumption patterns that may not directly translate to the effect of adding a cup or two of green tea to a Western dietary pattern later in life, a distinction that researchers studying this area are generally careful to acknowledge even while continuing to find the overall body of evidence genuinely supportive of a protective association. Stepping back from these 5 specific foods, it's worth addressing something important about how this entire body of research should be understood and applied, because misunderstanding this point is where a lot of confusion around cancer prevention diets tends to originate. None of these foods, individually or even combined, function as a guarantee against cancer, and no legitimate cancer research organization frames them that way. Cancer risk is influenced by an enormous number of factors, including genetics, age, environmental exposures, infections, and yes, diet and no single dietary choice can override every other risk factor a person might carry. What the research does support, with genuine and substantial evidence, is that these foods, particularly when consumed regularly as part of an overall dietary pattern, rather than as occasional additions, are associated with measurably reduced risk for specific cancer types, through biological mechanisms that laboratory research has helped clarify, and that make genuine scientific sense rather than representing coincidental statistical associations. This distinction between meaningful risk reduction and guaranteed prevention matters enormously for how this information should actually be used. The strongest and most consistent finding across cancer prevention nutrition research overall is not really about any single food in isolation, but about overall dietary patterns. Diets rich in a variety of vegetables, fruits, whole grains, and foods like the ones we've discussed today, combined with limited intake of processed meat and excessive alcohol, along with maintaining a healthy body weight and regular physical activity, collectively show some of the strongest cancer risk reduction associations found anywhere in nutritional research. The 5 foods we've walked through today represent particularly well-studied and evidence-backed components worth specifically prioritizing within that broader pattern, not because any one of them works in isolation, but because the cumulative weight of research behind each of them across both population studies and laboratory mechanism research gives genuine confidence that regularly including them in your diet is a meaningful, evidence-supported contribution to reducing your own cancer risk over the course of a lifetime. It's also worth addressing how these foods are prepared and consumed, since research has found that preparation method genuinely affects the protective compounds we've discussed throughout this conversation, sometimes in ways that run counter to common assumptions. With cruciferous vegetables, research on the enzyme responsible for converting glucosinolates into their active protective forms has found that this enzyme is heat sensitive, meaning heavy, prolonged cooking can reduce the amount of active sulforaphane and related compounds generated compared to lighter cooking methods like steaming for just a few minutes, or even raw consumption in the case of foods like coleslaw. Some research has found that pairing cooked cruciferous vegetables with a small amount of raw cruciferous vegetable, or with certain other ingredients like mustard seed, which contains a related enzyme, can help restore some of this activity even after cooking. An interesting and genuinely practical finding for anyone looking to maximize the protective compounds from foods like broccoli or cauliflower in their regular cooking. Storage and freshness matter across several of these food categories as well. Research on berries has found that polyphenol content can degrade somewhat over storage time, though frozen berries, contrary to what some people assume, generally retain their protective compounds quite well, since the freezing process itself, when done shortly after harvest as is standard for most commercially frozen produce, preserves these compounds more effectively than the several days of shelf-time fresh berries often experience before reaching a consumer, meaning frozen berries represent a genuinely evidence-supported and often more affordable and convenient option rather than an inferior substitute for fresh. For green tea specifically, research on brewing method has found that water temperature and steeping time affect catechin extraction, with very high water temperatures, at or near boiling, actually degrading some of the beneficial catechin content compared to slightly cooler water in the range, typically recommended for green tea specifically, alongside steeping times of a few minutes, generally extracting a meaningfully higher concentration of these compounds compared to very brief, steeping, practical details that matter if the goal is maximizing the research-supported compounds within each cup. It's also worth acknowledging a broader point about how nutrition research like this should inform daily decisions without becoming a source of anxiety, since that outcome would run counter to what this evidence is actually meant to support. None of the 5 foods discussed today need to be consumed in extreme quantities or with rigid daily precision to derive meaningful benefit. And the research generally reflects patterns of regular, moderate, sustained inclusion within an overall varied diet, rather than any single food eaten to excess. A few servings of cruciferous vegetables across the week, tomato-based dishes incorporated regularly, fatty fish eaten a couple of times weekly, berries added to breakfast or enjoyed as a snack, and green tea as a regular beverage choice, together represent a realistic and genuinely evidence-aligned pattern, one that fits naturally into an ordinary life, rather than demanding the kind of extreme dietary overhaul that research on long-term adherence has repeatedly found difficult for most people to sustain. And sustainability ultimately is what allows any of these protective associations the years of consistent exposure the underlying research actually measured.